Great, thank you for that introduction. Before I start, I just want to give a special thanks to all of the organizers for putting together this platform for us so that we're all able to share our exciting work during these challenging times. So for a little bit more background on myself, I've lived in Michigan my entire life, spent a lot of time at Lake Michigan. I am a fourth generation Spartan and will bleed green and white forever and always. I'm following both my uncle and my great uncle in choosing a career in fisheries science, although I did sort of fall into this field when I originally joined the Aquatic Animal Health Lab as a freshman. I was looking for veterinary research experience, but once I was introduced to the world of fish health, I was hooked. And while much of my time has focused on viruses, I do have interests in all aspects of fish health, but particularly those relating to conservation, population management, and One Health. I also have a very large, very hairy companion whom I normally love to show off, but today I'm hoping he continues napping for the next half hour. So jumping into my talk, as you're all aware, fisheries and aquaculture play a crucial role in feeding millions of people around the world. And while the yields from capture fisheries have plateaued over the past 30 years, global aquaculture has been increasing exponentially. And of course, with an increase in such intensive rearing comes an increased risk of infectious disease. According to the USDA National Agriculture Statistics Service, in 2018, the US produced almost 80 million trout, not including those that were raised for conservation efforts. But of those 80 million fish, only 63% of them survived to be sold. And of those 29 million fish that were lost, 92% of them were lost to disease. Now, many of these losses can be attributed to bacterial infections, and more specifically, flavobacteria infections. One of the most concerning aspects of these infections is that we know there is strong evidence of vertical transmission, both intra-ovum and with reproductive fluids, of at least one flavobacteria species, that being Flavobacterium psychrophyllum, which is the causative agent of bacterial cold water disease and rainbow trout fry syndrome. And now there's growing body of evidence that other flavobacteria may be transmitted in a similar manner. Unfortunately, we still know very little about these undescribed flavobacteria. Some might be pathogenic and others not. Established treatments may work against them, or they may not. What we do know is that some of these emerging flavobacteria can cause mortalities, but we have little knowledge beyond that. Based off of what we do know at this time, we think that there are actually a number of flavobacterial species contributing to early life stage losses in farmed rainbow trout. And with this hypothesis in mind, we put together a study designed to fill several important knowledge gaps. We opted to tackle this study in multiple phases. The first two focused on collecting and identifying the flavobacteria that are present in rainbow trout rearing facilities across the country. For phase 3, we wanted to understand what would happen if we experimentally challenged those early life stages with some of the recovered flavobacteria isolates. Now, the primary focus of today's talk is going to be on a portion of phase 4, but in order to establish a bit of context, I'm going to briefly walk you through what has led us to this point. In order to accomplish phase 1, sampling of rainbow trout occurred at 6 different hatcheries and private aquaculture facilities across the country. 1 in Idaho, 1 in Minnesota, 2 in Michigan, 1 in Ohio, and 1 in Pennsylvania. At each hatchery, over the course of a rearing cycle, samples were collected from the source water prior to entering each rearing unit, as well as the water exiting the rearing units. Samples were then collected from broodstock, unfertilized eggs, embryonated eggs, sac fry, and swim-up fry, which resulted in a total of more than 5,000 samples being analyzed. All water, broodstock, and fry samples were processed using our optimized flavobacterial screening protocols. Eggs were iodophore disinfected following routine hatchery practices, and an additional subset of eggs were disinfected at 4 times the recommended concentration. All samples were then inoculated onto multiple flavobacteria selective media and incubated at multiple temperatures. Following purification, each isolate was cryopreserved as part of our ever-expanding flavobacteria library. Eventually, a stretch of the 16S rRNA gene was amplified from each isolate, submitted for sequencing, and phylogenetic analysis performed. In total, this study resulted in 41 Chrysobacterium isolates and 490 Flavobacterium isolates. After performing a neighbor-joining phylogenetic analysis, these isolates sorted into more than 50 Flavobacterium taxa and 14 Chrysobacterium taxa. And if you haven't ever seen a printed out but legible version of a tree this size, I'll just say that we could have multiple people reading over it while still obeying the current social distancing guidelines. When we delve into these taxa a bit closer, we see that some of these isolates clustered closely with some known pathogenic flavobacteria, such as Flavobacterium psychrophyllum. We also recovered other known flavobacteria that have recently been connected to fish disease outbreaks, such as Flavobacterium plurixtorum and Flavobacterium spartansii. However, most of the isolates were distinct from all described species and may represent novel taxa. We had some clades, such as that containing the plurixtorum isolates, which were found to be widespread across multiple facilities. And in fact, these isolates were found across the country in samples collected from all 6 locations. In contrast, we also recovered isolates that were found at only a single facility. Adding additional complexity, we identified some isolates in broodstock and in eggs and in fry, while others seemed to be primarily life stage specific. And finally, we had over 100 isolates that were recovered from eggs after they'd been disinfected with the recommended iodophore dose. We also had more than 50 isolates that were recovered from eggs that had been disinfected at 4 times that concentration. Analysis of this sizable library of Flavobacteria isolates has yet again shown how complex rainbow trout-associated Flavobacteria are. We identified several clades containing isolates recovered from multiple life stages, which is suggestive of parent-to-offspring transmission. And it's now clear that Flavobacterium psychrophyllum, Branchiophilum, and columnare are not the only players we have to worry about. For the next phase of our study, we wondered if these Flavobacteria were specially equipped to resist innate egg defenses. In fact, it's already been shown that Flavobacterium psychrophyllum is resistant to both lysozyme, a naturally occurring bactericidal enzyme present within trout eggs, and whole trout egg contents. With that in mind, we set out to determine if the isolates recovered in this study were susceptible to lysozyme. And if you attended the Eastern Fish Health Workshop in Lake Placid, you may remember Chris Knupp's talk on this experiment. Briefly, in parallel, we inoculated 524 isolates onto flavobacterial growth agar plates and plates supplemented with 100,000 units per mil of lysozyme, which is equivalent to the level present in trout eggs. Of those 524 isolates, 5 of them were susceptible to lysozyme. The other 519 were resistant. So lysozyme alone clearly isn't preventing flavobacterial infections, but maybe there's a component or combination of components within the egg that's capable of doing what lysozyme couldn't. So to test this, we aseptically extracted whole egg contents from approximately 3,000 unfertilized rainbow trout eggs, into which we then inoculated the flavobacterium isolates and recorded viability afterwards. We tested a total of 515 isolates. Of those, 11 failed to grow in the presence of egg contents. So instead of happy eggs that are capable of stopping flavobacterial infections all on their own, we're left with this. And knowing that flavobacteria are present in the parents and are well suited to surviving within the fish egg environment, this egg stage is where we as fish health professionals have the most crucial ability to intervene. Vertical transmission of fish pathogens, both intra-ovum and in reproductive fluids, is not a new concept and is the reason that egg surface disinfection protocols are so widely implemented in aquaculture operations worldwide. Unfortunately, current disinfection protocols are inconsistent to ineffective. And in order to address this issue, we designed the following study with an ultimate goal of making improvements to current fish and egg treatment and disinfection protocols against flavobacteria. This study began by using cryopreserved stocks and our phylogenetic analyses to select 65 isolates from more than 30 clades, which were revived and diluted to a standard optical density. These isolates were recovered from water, broodstock, eggs, and fry, and included flavobacteria, chrysobacteria, and representative reference or type strains from both genera. Concurrently, working concentrations of our disinfectants were prepared. For this study, we chose 4 surface disinfectants that are already approved for use and are either used for egg treatments or used to treat at least one flavobacterial disease in fish. We opted to assess these doses as they're either equal to or flanking those that are currently being used for approved treatments. These 4 disinfectants at these concentrations were chosen not only to guide in developing improved egg disinfection protocols, but also to solidify our expectations for the therapeutics already in use treating flavobacterial disease outbreaks in production fish and broodstock. We then took our flavobacteria isolates and exposed them to the 14 disinfectant doses for 3 different time points, after which samples were serially diluted, drop plated, and the colonies counted. Using the colony counts, percent reduction was then calculated for each disinfectant dose at each time point for each isolate. In order to present these results, I have a series of graphs that are all set up like this, with the disinfectant and dose listed at the top, incubation time on the x-axis, and percent colony reduction on the y-axis. So essentially, with an effective disinfectant dose and duration, you would see all of the isolates cluster at the top of the chart. Additionally, I have the different isolate groups designated as Chrysobacterium with the green circles, Chrysobacterium type strains with the black plus signs, Flavobacterium as orange diamonds, and Flavobacterium type strains as the black X's. As you know, depending on water temperature, formalin treatments for parasite control range from 150 to 250 parts per million. When we tested the low end of this range, we did see some reduction, but in a lot of isolates were still surviving, even out at 30 minutes. And it wasn't until we increased both the exposure time and the dose up to 1,000 to 2,000 parts per million, which is the recommended dose range for eggs, that we saw substantial reductions in growth. One interesting finding here is that it appears that the Flavobacterium are quite a bit wimpier in the face of formalin than the Chrysobacterium, which begs the question, by implementing these formalin treatments, is it possible that we are inadvertently selecting for Chrysobacteria? One of the recommendations for treating bacterial gill disease in salmonids is 100 parts per million hydrogen peroxide for 30 minutes. However, at this dose, we once again saw a range of reduction across the 3 time points. Even with the recommended 3 treatments performed every other day, you're unlikely to see a sufficient bacterial reduction for many of these isolates. And as more and more people are seeing, there are emerging flavobacteria that can be contributing to bacterial gill disease. So it's important to acknowledge that they're not all equally susceptible to the compounds that we're using to treat. When we increased the dose of hydrogen peroxide to the range recommended for treatment of eggs, we did finally see complete or near complete reduction in growth after 30 minutes. Unfortunately, the duration of egg treatment is only recommended up to 15 minutes. And at these lower times, we were still seeing a significant proportion of isolates with less than 80% reduction. With chloramine T, for the first time, we saw substantial reduction in growth right from the start, with no growth at 10 or 30 minutes, even at the low end of the recommended dose range for treating bacterial gill disease or columnaris disease. In fact, there were only 2 isolates that showed any growth at all when exposed to 20 mg per liter. Both of these were Chrysobacterium, one of which was isolated from milt and the other from water. Complete reduction was seen for all isolates at 40 mg per liter, which, as a reminder, is actually twice the high end of the recommended dose range. But overall, this is good evidence that INAD use of chloramine T should be effective. At least when it's one-on-one, pathogen versus disinfectant, it looks very effective, even at significantly shorter durations than what's currently being used for treatment of flavobacteriosis, which is a good first step in understanding how these treatments may function in vivo and under field conditions. With iodine, we had complete and total reduction in growth with all isolates at all concentrations and all time points. And this was a bit surprising to us because several of these isolates were recovered from eggs that had been disinfected with 100 parts per million iodine and several from eggs disinfected with 400 parts per million iodine prior to sampling. What this means is that these bacteria, while extremely sensitive to iodine in a sterile laboratory environment, are more than capable of evading the disinfectant when either on or inside an egg. This implies that the inconsistencies with disinfection lies not in the chemical, but with the protocol and its application, which is a good thing, because it means that iodophore disinfection, if a proper protocol can be established, has the potential to significantly reduce vertical transmission of flabobacteria. As I just mentioned, we saw a stark contrast between the in vivo and in vitro effects of iodophore disinfection, which suggests that if we were to modify our standard disinfection protocols, they could become more efficacious. With formalin, we saw that it wasn't very effective at the low concentrations typically used in production fish and broodstock, yet it was pretty effective at the higher doses used to treat eggs. However, in using this chemical, are we selecting for Chrysobacterium on our eggs? I don't know the answer to that, but it's certainly an important question to consider. In contrast, chloramine T was really quite effective even at low concentrations and shorter exposure times, and depending on the weight you give an in vitro study, shows promise against a wide range of flabobacteria. So, taking a step back and combining these results with the findings from the larger study, we now have more evidence than ever of the substantial diversity of flabobacteria associated with rainbow trout farming across the country. As we continue to uncover more and more about this vast family of bacteria, we are realizing that there are a large number of both described and novel species contributing to disease outbreaks, particularly in the early vulnerable life stages. These findings have significant implications for disease prevention, as we demonstrated that not only are our current egg disinfection methods inconsistently effective, but use of these, as well as other topical therapeutics, may in fact be unintentionally directing the flabobacteria communities toward more resistant species. Even without knowing the mechanism, it's clear that genetic diversity does matter for some disinfectant compounds, as was evidenced by the variation in success of formalin and hydrogen peroxide. In order to halt and/or reverse these shifts and prevent future disease-related losses, improved chemical treatment protocols must be developed, particularly those that are able to interrupt parent-to-offspring transmission. The findings of this current study strongly suggest that it is the protocol rather than the compound that is most responsible for past disinfection inconsistencies. And were we able to dial in and establish more effective prevention and treatment protocols, this would be a powerful tool to use in the fight against pathogenic flabobacteria in the trout industry. And with that, I would just like to graciously thank the USDA NIFA for funding this project. Again, thank you to our seminar organizers for putting this together. And thank you to all the facility managers, as well as the numerous past and current Aquatic Animal Health Lab members who have lent their support to the study. And now I can take questions.
Great, thank you very much, Megan. That was an excellent talk. If you have questions, feel free to unmute yourself, or you can type them in the chat box and I can read them to Megan.
Hey, Megan, how are you? This is Esteban from East Davis. Hello?
Hi, yes.
Do you hear me? Okay.
Yep.
So I have a couple of questions, in terms of the method that you use. So it is all related to the testing of the disinfectants in vitro. So a couple of things that I missed, what was the concentration of bacteria that you were suspending, and in what type of media were you suspending this before testing?
Yeah, so after the isolates were all revived on solid media, we then subcultured them into broth media, let them grow for 24 to 48 hours, and then we resuspended those in sterile water and diluted them to an OD of 0.1, and then took that, and so then all of our disinfectants were also diluted in sterile water as well. So then when we combined those, that's what they were diluted in.
So because again, so I don't know what is the OD translation, to CFUs per ml. Do you know that on the top of your head or not?
I don't,
no?
Sorry.
Yeah, no, that's fine, because when we test, for example, antimicrobials, one of the key limitations or the key issues that you have to always standardize is CFUs per ml, so that you are not overloading the system to the point where pretty much anything will be seen to be resistant. So standardizing that, typically for antimicrobials, you test it to around 10 to the 5 CFUs per ml, before final testing. So that is one thing that would be good to standardize. And then the other one is also, because we have played around, with other bacteria in these similar scenarios, is using sterile media or sterile water for testing. And, one of the issues is that, again, some of the components, present in the water, and even sterilization of the water can mess up, some of the ions that are suspended in the system. So probably it would be good, to test at least some of them using filter sterile, like hatchery water, so that all of the components, that again, except the microorganisms, that you have, from the hatchery water are actually used when testing, the disinfectant, because that will be the best way to equalize, what you're testing in the, in the in vitro to what it is seen in vivo. And then lastly, my last point, with the iodophores, that is something also that I think you guys have explored in your laboratory, but I cannot remember if you guys are continuing to doing that. I know that in another project, Tom is looking at it is the biofilm formation. And if I recall, some of these eggs, some of these bacterium are capable of forming biofilm in the egg. And because you are testing with the method that you just described, you're testing the planktonic form of the chrysobacterium, those guys probably are more susceptible to the disinfectant than the biofilm. But in the egg that you are getting the bacteria from, most of those bacteria probably are in the biofilm form. So probably that will be a way to also explain, might not be the iodophore are really good. It might be still that you are dealing with a different form of the bacteria in form of the biofilm, and that's why you're seeing the higher resistance at the 400 ppm for iodophore versus the planktonic one. So I don't know if Taylor is in the online, but Taylor has been exploring some of these with Streptococcus. And if I recall, a similar thing happened with Streptococcus ine, where the higher concentrate, the biofilm basically made iodophore useless, whereas the planktonic form was still susceptible to.
Yeah.
So just bear in mind a couple of those, as you write the papers and whatnot.
Yeah. Hi, Megan. That was a really good talk. But yeah, basically, planktonic forms were super susceptible when I made sure to keep them agitated so they couldn't form a biofilm. But when I purposely formed a mature biofilm, that was super resistant against the iodophore.
Neat. Thanks.
Yeah. So Esteban, and excellent points. So yeah, you're exactly right. This was sort of our initial starting point, and I completely agree with you. That I do suspect that the biofilm, the ability to form biofilm is a big role player, as is presence inside the egg. So you're aware of the studies with the group from Japan that have shown that if you water harden eggs, if you disinfect eggs in iodophore prior to fertilization, so isotonic solution, that it actually seems pretty effective at getting rid of flavos. So I'm dead on with, I think you're exactly right, that it's either biofilm or presence inside the egg, but some component that's very different in the real world versus in a petri dish is explaining. But I think where we were most excited about these results is the fact that, if it was the opposite, if we did this testing with iodophore and a bunch were resistant in a petri dish, then that you need to turn your attention to a different compound. If it can't even work under those conditions, then you need to find something else. So
yeah, and I'm curious also, Megan, and Tom, as you guys deal with potential a lot of them, being vertically transmitted. I'm curious, if some of the management protocols, should be really targeted, to the broodstock. And I don't know. Again, I hate to say treating, prophylactic treating with antimicrobials,
Mm-hmm.
because there's so few that we have available that can be used, but potentially immunostimulating the broodstock, or vaccinating, again, it will be ideal, but against the chryseals and the flavels being so diverse, I don't know. I know that there is a live vaccine out there, that has been shown some cross protection, but I think it will be interesting, to see what will happen, if you, immunostimulate the broodstock, and if you're able to reduce, the overall numbers of some of these chryseals also in the egg, and eventually the mortality of the young fry.
Yeah,
Have you guys looked into that?
Not that I'm aware of. I think with this project specifically, where we're kind of going more toward is altering those management practices in terms of how these compounds are used, as kind of a starting point, seeing if we can go from there. But I think those are great ideas. I would like to avoid prophylactic treatment per se,
Absolutely.
as much as absolutely possible. But any other options we have that can keep those broodstock from passing on these bugs, I think are great ideas.
Yeah, I mean, I'll be, yeah, like the anti, again, that is useless also, against the chryseals, because they're so resistant to everything. But I think it will be interesting, immunostimulation, for example, and I don't know if it has been looked at into this area, into potentially passive immunity into the eggs, trying to get, because again, if you're seeing most of these bugs already in the broodstock, and a very similar, microbiota passed, into the eggs, then potentially, just stimulate as many of these mucosal, antibodies, and stimulate a little bit of the innate immunity that is passed on, and try to see if you can convert more protection into the early life stages as they produce their own or develop their own immunity.
Yeah.
Great job, Megan. Very cool. Very cool.
Thank you.
Those dendrograms are so scary.
So Esteban, just really quickly, the Ken Kane's group has looked a little bit into, I think they were using beta glucans. It wasn't in the context of protecting brood stock and then protecting the offspring, but they showed that I think it was the beta glucans did seem to have some protective effect, but that eventually plateaued. So there was a risk where I think after a certain point, I forget, but I want to say 2 to 3 weeks after being fed this, that the fish actually became, it was either more susceptible or at least equally susceptible. So it seemed like the protection had a short duration and you could overtax it if you kept it going with it.
Yeah, you have to play around it with it, but again, the point is if you just prior to, if you just basically try to immunostimulate as much as possible prior to responding, I don't know.
Great conversation. Any other questions out there?
Yeah, this is John Drennan. Can you hear me?
Yeah.
Sure can.
Hey, it's complicated. I don't want to play a blame game at all, but I mean, it'd be interesting with all the egg source. How many of the egg sources were from Trout Lodge? And obviously, I don't want to go down that path, but I mean, it might give peace of mind if we know. I mean, I'm sure all the states are getting eggs from Trout Lodge for part of their trout rearing. Have you looked at? Well, my question is, how many are from Trout Lodge? And maybe if you could do something like this, it might give peace of mind of what are in those eggs, when it comes to flavos and chryseals. Because, I mean, we have hatcheries that pop up like clockwork with cold water disease. It could be in the water already, biofilms in the system, but it also could be from the eggs.
Yeah, we're actually not sharing we're keeping the identities of the actual locations we
totally
spend.
Yeah.
Sounds good. Nice try, John.
Yeah.
Yeah.
John, that's an excellent point. And I think I can at least, I don't want to steal your thunder, Megan. But I think I can at least say that your concerns are right. But I wouldn't, based on our data, I wouldn't say that there's at least for these oddball flavos, I wouldn't say that there's a smoking gun per se of where they're necessarily coming from. I think there's a lot of them in a lot of different brood. But you're right. That's something that people have to be worried about, is that some of them could be coming from a common source.
Yeah, I agree with you. And really it is manageable. It's just takes effort and costs money. That's all.
All right. Well, thank you very much, Megan.